As part of developing the CapScan gut sampling device, we recorded the microbiome in action and documented the sampling of the CapScan device using capsule endoscopy. The first part of this video shows an apple being digested. The process starts with mechanical breakdown of the apple by the teeth. The apple is swallowed and moves from the esophagus into the stomach. In the stomach, hydrochloric acid and pepsin start to break down the proteins in the food. Waves of peristalsis break down the apple into acidic chyme. The chyme is kept in the stomach by the closed pyloric sphincter. The pyloric sphincter opens when the small intestine is ready to process a bolus of chyme. Now we are in the duodenum. The pancreas, gallbladder, and liver release digestive fluids such as bile salts, peptidase, lipase, and bicarbonate to neutralize the acidic pH of the chyme. The digestive juices are mixed with the food in the duodenum to emulsify the fats and prepare the nutrients for absorption by the villi of the small intestine. Now we're in the jejunum, where most of the nutrients are absorbed. Triglycerides undergo enzymatic hydrolysis into fatty acids. Simple carbohydrates are broken down into sugars and absorbed. Here, gut microbes start converting bile salts into primary and secondary bile acids. Gut microbes are also activating and metabolizing vitamins and drugs. Now we've reached the ileum, where the vast majority of bile acids are reabsorbed into the portal vein and returned to the liver. The liver reintroduces the bile salts into the duodenum. The body recycles and reuses bile acids multiple times, even during the course of a single meal. We've now crossed the ileocecal junction and are inside the cecal portion of the ascending colon. Microbial densities increase exponentially relative to the small intestine. Half of the luminal contents you see here are bacterial cells, dozens of species coexisting in complex cross-feeding networks. This is the highest concentration of microbes of any known ecosystem, separated from our blood vessels by a mucus layer that is just a fraction of a millimeter thick. Fermentation of complex carbohydrates by bacteria creates short-chain fatty acids that feed the cells lining the colon and regulate the immune system. The molecular patterns on the surface of these bacteria also regulate the immune system. After several hours, when the next meal is eaten, the contents of the ascending colon are transferred via peristalsis to the transverse colon. In the transverse colon, packets of stool are formed and transported to the left or descending colon. At this point, there is much less biochemical activity. In the descending colon, most of the water in the stool is reabsorbed, and the stool is prepared for a bowel movement, eventually ending up in the toilet. We also documented the sampling of the CapScan device by physically attaching it to a video capsule. Here, we are sampling the jejunum with the luminal contents entering the device at the location of the white arrow. A second CapScan device is now traversing the entire small intestines and entering into the ascending colon with semi-formed stool. The white arrow shows the fluids of the ascending colon entering into the CapScan sampling device.